Assalamualaikum warahmatullahi wabarakatuh. My name is Asif. There, I will to explain my presentation about genome. Recent advances in DNA sequencing technologies have made available an enormous resource of data for study of bacterial genomes. The broad sample of complete genomes currently available allow us to look at variation in the growth, features, and characteristics of genomes, while the detail of the sequence reveals some of mechanisms by which this genome evolved. Annual review of genetics include an outstanding review by Sherwood, Chess James, entitled The Diverse and Dynamic Structure of Bacterial Genomes. This current review aims to update some of the ideas covered by questions. Indeed, it is fascinating to see just how rapidly this field has advanced in such a short time, largely due to the surge in whole genome sequencing and the associated wealth of new data over the period. By end of 1998, 17 complete bacterial genome sequences were available. By the end of 2003, prior to the advent of whole genome sequencing, our knowledge of prokaryotic genome structure was largely limited to what we could determine from polycrystalline gel, electrophoresis, and classical genetic maps. From the early 1970 through to the 21st century, rapid advance in the technology associated with DNA sequencing mean that obtaining the complete DNA sequence for a bacterial genome went from being very challenging to relatively simple, at least for certain genomes. By the end of 2003, the public sequence database held the complete sequence of 99 bacterial and 12 archaeal genomes covering 69 and 12 different species, respectively. This represents an unprecedented dataset with which to investigate prokaryotic genome structure. Studying each genome in isolation has brought novel insight, and this new knowledge has often been further enriched by comparison of genomes from related strains. Next, genome structure. The obvious parameters for describing a bacterial genome are size, geometry, replicon number, and G plus C content. Variation in this alone ensures that there is no such thing as a typical bacterial genome. Actually, defining a bacterial genome is complicated by the variety of DNA replication found within bacterial cells. This allows us to dis- disregard transit replication, such as plasmid, but the definition of chromosome is itself controversial. Next, genome size. Prokaryotic genome size vary across more than 20-fold range. Look at figure 1a. Figure 1a show how this size range lies relative to all forms of life, overlapping with the small eukaryotic and the large virus. It is now clear that archaeal genomes, previously thought to cover a relatively narrow range, vary to similar degrees of eubacteria. Within the prokaryotic group, different phyla cover broadly overlapping size range. Look at figure 1b. Even within species large-scale variation can be seen. Genome of and Sericea coli, Prochlorochogus marinus, and Streptomyces follicular have all been seen to vary by more than 1 million. Bacterial genome size in bacterial genome size is the sum of different genetic events, such as gene duplication, horizontal accretion, and ligand-specific gene loss, and therefore not a good indicator of evolution lineage. Although distantly relates, the Proteobacteria and Actinobacteria both appear to reach the extremes of bacteria, suggesting that the factor limiting genome size might be common to all. The exact nature of this limiting factor is unclear. The minimal genomes concept is attracting great interest, with the seductive aim of defining the minimal set of genes required for cellular life. So the minimal set will of course vary depending upon the needs. Definition of a maximal genome is perhaps less intuitive, whereas a minimal genome is limited by the ability to live. The maximal boundaries are more difficult to draw. Complex issue routing get to generation time, replication rate, and energy supply need to be considered, as well as simple practical issues such as the physical space taken up by the genome within the cell cycles. Okay, conclusion and future perspective. Original lack of bacterial genome structure has clearly benefited from the recent advance in genome sequencing. We now have a we now have detailed information relating over to 150 bacterial genomes, and this figure continues to rise. This large database helps draw a useful picture of bacterial genomes in terms of size, geometry and replication number, and the variation within these parameters. The detail of the data has brought new insight into genome structure and evolution, and has provoked many hypotheses to explain observed patterns. The unknown now is for those hypotheses to be proved experimentally, so in many cases they may simply be strengthened or weakened by the accumulation of further genome sequence of the future genome sequence. Science genome sequencing required considerable funding. The focus of bacterial genomics thus far, thus far has been motivated primarily by medical and commercial consideration. The priority has been to sequence the genomes of pathogens along with a handful of model organisms and important environment species. This strategy has greatly advanced our understanding of the mechanism by which bacteria evolve and adapt to best fit their needs, and for pathogens affect immune response. The prioritization of research resource has inevitably meant that the current dataset is biased, but continued sequencing and decreasing codes should help us move toward a representative view of all bacterial genome regardless of our ability culture them. The emerging field of metagenomics, the analysis of all genomes within a niche, will be crucial in this endeavor. Recent studies have carried out random sequencing of DNA representing ecological communities from an acid mine drainage site, and from Sargasso Sea. The dominance of just a few species within the EMD ecosystem means that two near complete and their partially complex genomes could be constructed. The surface sample was much more complex, so the billion base of sequence generated did not assemble into many whole genomes, but did indicate the presence of over 1,800 species. These are the first step for metagenomics, but if the progress of genomics is anything to go by, we can expect to see hundreds, if not thousands, of the complete bacterial genomes in the next decade. Next, please. Okay, thank you for time and wassalamualaikum warahmatullahi wabarakatuh.